Hi there, iWatchers! Welcome to iBio Academy, where we talk about the real natural science and no cap. And for today's episode, the Calvin cycle in photosynthesis. Have you ever wondered how the world works, or how is life even possible in our planet? Well, let us introduce ourselves first. We are the third group of STEM 12 Michael Faraday, and let's talk about the Calvin cycle.
A quick overview of the Calvin cycle. The Calvin cycle represents the most sustainable production lines. It is known as the light-independent reactions or dark reactions of photosynthesis. Carbon molecules from the air are absorbed by the Calvin cycle and converted into plant matter. Its job is to make 3 carbon sugars, which are then used to make different sugars including glucose, starch, and cellulose.
Now let's talk about the processes of the Calvin cycle. There are 3 key phases in the Calvin cycle. The first one is carbon fixation, the second is reduction, and the third one is the regeneration of ribulose diphosphate.
Carbon fixation is the process by which inorganic carbon is added or fixed to an organic molecule, hence the name. In this phase, the carbon dioxide molecule is attached to a 5-carbon sugar molecule named ribulose diphosphate RUBP. Aided by an enzyme called rubisco or RUBP carboxylase, rubisco is believed to be the most abundant protein in the chloroplast and maybe on Earth. The resulting product, a 6-carbon sugar, is extremely unstable and immediately splits in half. The split forms 2 molecules of 3-phosphoglycerate or 3-carbon.
After carbon fixation, hence the name reduction, this stage utilizes the ATP and NADPH created in the light-dependent reactions of photosynthesis, which are used to convert the 3 PGA molecules into molecules of a 3-carbon sugar, glyceraldehyde 3-phosphate, or G3P, where ATP is converted into ADP and NADPH is converted into NADP+. Then they will be transported back to the thylakoid membrane and will enter the light reaction.
The regeneration of RUBP comes after the reduction. 5 molecules of RUBP will undergo a series of complex enzymatic reactions involving numerous steps in order to regenerate 3 molecules of RUBP. 3 molecules of ATP are required to provide another set of RUBP, allowing the cycle to start and continue again.
The products of the light reaction, ATP and NADPH, as well as carbon dioxide, are the materials that are needed to begin the Calvin cycle. Each material has its different function. From the air, carbon dioxide is transformed into sugar. The Calvin cycle produces high-energy sugars by using the energy in ATP and NADPH. Aside from that, chemical reactions are also fueled by the energy provided by ATP and NADPH.
The Calvin cycle mainly produces a 3-carbon sugar known as G3P or glyceraldehyde 3-phosphate. The cycle needs to spin 3 times in order to make 1 molecule for G3P. For the final products, the Calvin cycle produces glucose.
You may be wondering how glucose was created. I provide you with some more information about it. Each turn of the Calvin cycle fixes 1 molecule of carbon that may be utilized to produce sugar with each revolution. 1 molecule of glyceraldehyde 3-phosphate requires 3 Calvin cycle spins to be produced. 2 molecules of glyceraldehyde 3-phosphate can be joined after 6 cycles of the Calvin cycle to form a glucose molecule. Another interesting fact is that Calvin cycle was discovered and named after Dr. Melvin Calvin, an American biologist, in 1957.
A quick recap of our discussion. We now have learned the basic background information or overview of the Calvin cycle, such as its alternative term called dark reactions and how it takes place in the stroma. We have also learned the 3 necessary stages or steps that occur within the Calvin cycle, such as the carbon fixation, reduction, and regeneration of RUBP. And lastly, we have also learned the necessary materials that are needed in order for the process to take place, as well as the products that are eventually made during this process.
And that is the metabolic process, the Calvin cycle. Now, does life on Earth seem more possible? Or are there still wonders and doubts inside your head? We hope that this video was limpid and informative enough for your liking. That could be all. See you on another episode of iBio Academy.